Thanks for joining this afternoon. My name is K Warma, CEO of ProMIS Neurosciences, and thanks to the Burton folks for the invitation again this year. Great to see the enthusiasm in the crowd, and it's been an interesting year for us. I'll take you through a little bit of the storyline, but we're excited. It's been especially productive few months. ProMIS Neurosciences, I'll kind of go through a bit of an operational update here, just as a bit of a way to remember us. ProMIS is based on our platform technology, which is based on protein misfolding. So a little bit of a play on words, ProMIS. But our founder, Dr. Neil Cashman, is one of the leaders in protein misfolded diseases. We are a publicly listed company, so I'll just remind you to all please look at our SEC filings just to get a true understanding of our operations. So ProMIS is a publicly listed company. We're traded on the NASDAQ Capital Markets. Ticker is PMN. We are based primarily in Boston and Cambridge, but we do have a Canadian history. As I mentioned, Dr. Neil Cashman, our founder, our CSO, developed this technology, protein misfolding technology, out of the University of British Columbia. Our discovery lab is still in Vancouver, whereas most of us were pretty scattered, but when we meet in Cambridge. But we have a strong historical root in Canada. I'm Canadian. I studied up the road at University of Toronto. I have a neuroscience degree, so it's nice to come full circle with the neuroscience platform. So I'll touch on a few of the highlights here. The company is a clinical stage company, and I'll spend a few minutes talking through our clinical trial and our clinical candidates. We have our platform, and I'll come to this in a minute, is really based on our ability to design and create novel, selective antibodies targeting misfolded proteins. And that's really kind of our claim and our differentiating factor as far as the design of our antibodies. We have our lead asset is in the clinic. As I said, it's been a big year for us. It will be a huge year moving forward for us in 2026. We have data that reads out in a matter of months in Alzheimer's disease. So we are running a fairly robust phase 1b clinical study in Alzheimer's patients. As I said, there's an interim analysis coming up in a few months, and then final top-line results will come towards early next year. So significant milestones moving forward this year. We have, I won't really touch on the market expectations around an Alzheimer's product. We know there's a huge unmet medical need, even though we have a couple of products on the market. In Alzheimer's disease, the risk-benefit of those products are pretty poor. We have an outstanding team, in addition to Dr. Cashman, our founder CSO in Vancouver. Our chief medical officer is a practicing neurologist, Dr. Larry Allsteele. He's treated Alzheimer's patients all of his life, and he ran CNS at Pfizer. Our chief development officer ran CNS at Genzyme. So a really quality group of management here. Interestingly, we were just coming off the back of a really important financing. The one box we had not checked was, are we fully funded to data? And now we are. We closed the financing a couple of months ago, up to $175 million. And the syndicate that came together was superb. It was led by Ally Bridge and Janus. Wellington participated, Deep Track. So now we're funded kind of through the end of next year. So cash position is quite favorable. Just touching on the platform a little bit, a lot of this discussion we get around, well, how are we able to design these antibodies that are truly selective to a particular species of protein? And it really comes down to the differentiation between our platform and the conventional approach to designing antibodies. And just focusing on ours, and it's really the beauty of having the ability to predict how proteins are going to misfold. And we know that proteins are manufactured by the cell, and as they come out of the cellular machinery, you remember from your biology days, they're folded upon themselves in the three-dimensional form, this very elegant shape. And as they are folded properly, they go on to perform their function. But constantly or continually, these proteins are coming out of the other side of the cell, and they're misfolded or misshapen. But typically, your immune system grabs hold of these misfolded proteins and discards them. But as you get older, immune compromised, sometimes your immune system can't keep up with getting rid of these misfolded proteins. And if they're allowed to build up and aggregate, that's when you get many, many different diseases that are established because of these misfolded proteins. So we have the ability to predict how certain proteins are going to misfold. And I won't get into the details, it's a computational approach we have a chief physics officer in our company. So this is an algorithm that we apply, kind of like we were doing AI back before AI was cool, to be able to understand how proteins misfold. So it's a linear epitope, but it's also a conformational epitope, a three-dimensional epitope, to which we design an antibody, and that antibody selectively targets only this conformational epitope, which exists only on the misfolded form of the protein. So I'll come back to that in a minute because it's really important in differentiating us from the conventional way of designing antibodies. And because of that, we've built up a pipeline of candidates. Different diseases are driven by different proteins, misfolded proteins. So our target for Alzheimer's is amyloid beta, kind of a well-recognized Alzheimer's target. So A beta protein for Alzheimer's. The candidates coming behind that are misfolded forms of TDP-43 for ALS and misfolded alpha-synuclein for Parkinson's and Lewy body dementia. So we've got a really healthy pipeline of highly selective antibodies. So I'll touch on the Alzheimer's space since that's where we are in the clinic, and that's what the focus is certainly going to be on over the next number of months. So just kind of briefly in the scientific cascade of Alzheimer's disease, there's a whole, as you can imagine, a whole pathway that leads to different pathologies, if you will, all building towards Alzheimer's disease. And for us, I'm not sure if there's a laser on this or not. Doesn't really matter. But for us, you can see the PMN310 target. Our target, because there's multiple different species of amyloid beta protein, and we're specifically going after kind of this upstream in the cascade, these low molecular weight oligomers. And that's our target, upstream of plaque, upstream of tau. So our thinking is, and knowing that these oligomers are highly toxic to neurons, that's where our selective antibody is targeted, towards only these toxic oligomers that occur upstream in the pathogenesis of Alzheimer's disease. And you can see as you move down, as I mentioned, plaques will form through the aggregation of oligomers, which go to form protofibrils, which go to form plaque, and then they trigger the phosphorylation of tau. So we believe, given the toxicity of the oligomers and given the upstreamness, if that's a word, that's a relevant target for us, and others certainly believe that as well. And if you look at, and part of the challenge in Alzheimer's, as I mentioned, there's multiple different species of amyloid beta. We used to think, we the field, used to think that plaque was driving kind of disease progression in Alzheimer's. You all are familiar, I'm sure, with the buildup of plaque depositions, plaque formation in the brain. And that was supposed to be the target. You could image plaque, you could see it. It doesn't look nice, it's got to be harmful, it's got to be driving disease progression. We now know that's not true. There are drugs that completely eliminate plaque, and the patient continues to decline. So plaque is not the target, and I'll come back to plaque in a minute. We know there's the monomers, another form of amyloid beta, but that's a beneficial form of A beta protein, so you don't want to target that. And then you've got these toxic oligomers, which now the field is pointing at these upstream toxic oligomers that drive cognitive decline. And they're very low concentration versus the plaque and the monomers. But then the challenge is, how do you design an antibody that only hits the top of the pyramid and doesn't cross-react with all forms of amyloid beta? And that's the beauty of our platform. As I mentioned, the binding three-dimensional epitope only exists on oligomers. So we believe we don't bind plaque, and we certainly don't bind monomers. And we've done a ton of studies to pressure test whether or not we're binding plaque. And that's one of our claims, is we won't bind plaque. The other interesting thing with drugs that bind plaque, which is all of them out there, the two on the market and many in development, because they can't selectively target the oligomers. All drugs that bind and clear plaque are associated with a very serious side effect known as ARIA, A-R-I-A. And ARIA essentially is swelling and bleeding on the brain. You can kind of imagine a little bit of an analogy. You've got these kind of plaque deposits in the cerebrovascular structure, and if you bind and kind of pull them away, you're going to get bleeding and disruption of the vasculature. And that's what we see with the drugs that bind plaque. 20, 25, 30% of patients have bleeding and swelling on the brain. So not only is it not really responsible for driving cognitive decline, it's also associated with a pretty severe side effect issue. So our claim now is that we've got a drug candidate that targets only the oligomers. So all of our drug is directed towards the actual target. We're not binding plaque, so therefore we believe we will significantly reduce the incidence of ARIA, which is a big deal currently with the drugs on the market. So if you look at kind of the generation of antibodies over the, well, really over the years, and how they've been designed and developed, you have kind of solanezumab, which was designed kind of interestingly. They targeted these monomers. This is back years ago when they thought that monomers was the actual target. As I said before, we now know that monomers are actually a useful form of amyloid beta, totally the wrong target, and they show no clinical benefit. And then you come with the other antibodies, gantenerumab, which is now being repositioned and refashioned into trontinemab by Roche. And gantenerumab binds everything. So it got soaked up by the monomers, it got soaked up by plaque. So very little of the drug actually got to the toxic target. Showed no clinical efficacy. So we'll see what trontinemab does, probably similar. But then you get to the donanemab, which is Eli Lilly's drug. It's on the market. Lecanemab, which is Eisai's drug, Biogen Eisai, on the market. They're more selective. I mean, lecanemab was actually designed to exclusively bind the oligomers. But again, it cross-reacts. It's more selective, but it still cross-reacts with monomers and plaque. It's still associated with ARIA. So as you work through the, through the generations, if you will, of antibodies, you get more and more selective, and then we come to the clinic, and hopefully to the market, with what we believe is an antibody that's highly selective, again, but only for the toxic oligomers. And you can see on the side there the percent of ARIA that's associated with the drugs that have been developed in those other areas. So this is one study we did in-house, because as I said, we really wanted to pressure test whether or not we bound plaque, whether or not we had a differentiated asset, because it's really important. Well, it should be important for anybody. It's certainly important for me as a CEO to have a differentiated product that has a chance to be best in class. Because others have said they exclusively bind oligomers, and in the end, they cross-react and have ARIA. So we tried and tested. So the top row, these are brain sections from Alzheimer's patients. And they light up green, showing that there's plaque in these sections. And then the purple shows which antibodies actually bind to that plaque in the brain. And if you can see, the ones certainly on the right-hand side here, all fluorescing purple, all bind plaque. And that's lecanemab, donanemab, ACU193 from Acumen. The only one that does not bind plaque is PMN310, kind of third from the left. So this is something we did. We did a quantified analysis to say what's the level of fluorescent for these antibodies. And again, PMN310 on the left there, the only one that demonstrated no binding of plaque. So again, we were confident going into our clinic that we had something that was truly differentiated. And it was important to talk about it with the investors. It was important to talk about it with the investigators in the trial as well. And if you look at the two drugs in the market that I mentioned, again, the ARIA in the middle, this bleeding and swelling on the brain, is a huge challenge for them. The FDA has put a black box warning on each of those drugs, and the black box means extra careful attention because they've got a serious side effect issue. The efficacy on the right side, on the left side, you may not be able to see it, but it's kind of modest efficacy. These drugs have shown to be moderately efficacious in kind of 27% range of efficacy. So pretty modest efficacy, pretty serious side effects. So the risk benefit, pretty poor. Again, everything's a step in the right direction for sure. I don't want to beat them up too much because these are all advances which are helpful for patients. And at the end of the day, they're still predicting, $3-4 billion peak sales. So even with moderately effective, somewhat, safety issues, they've got a $3 billion a year drug, which also points out that there is commercial validation. There's kind of regulatory validation. So if we come or when we come with our drug that has a better risk-benefit profile, we'll be in good shape. The 1A study that we completed a year and a half ago, again, it was in healthy volunteers. It showed that the drug is safe and well tolerated. It also importantly showed that the amount of drug we had the amount of antibody we had across the blood-brain barrier was sufficient to fully engage our target. And that's important in the day of brain shuttles and such, that we've got enough drug across the blood-brain barrier. We don't believe we'll have an ARIA liability. So the phase 1A was, positioned us well for the 1B that we're running now. Probably the best decision we made as a company was in the design of the 1B study, where others have kind of done quick and dirty studies and a few number of patients looking at biomarkers. We took a step back and said, and I told the team, I want to have a study design, even though it's a phase 1B, so first in patients, I want to have a study that gives us a fairly definitive answer as to whether this antibody truly works. So it's got to include enough patients, and it's got to have a clinical signal. So the team came back and we designed this study. It's a placebo-controlled, double-blind, randomized study, 3 cohorts, 3 ascending doses. We wanted to target 100 completers. So we initially thought about enrolling 128 patients to end up with 100 completers, given the dropout rates of other Alzheimer's studies. The endpoints we're measuring are kind of a series of biomarkers, and there's a number of them we can touch on that are quite relevant. We're looking at safety. Obviously, the incidence of ARIA, the swelling and bleeding on the brain, is going to be definitive for us. And then we've also got the FDA clinical endpoints here, the CDR sum of boxes, ADAS-Cog. So it's a very robust study for a 1B study. We have completed enrollment. Enrollment's complete as of last December. And interestingly, as we were winding up the study, kind of getting close to our 128 target, the sites were very encouraged. I think they've been very encouraged with the study, given the potential for lack of ARIA for the first time. They kind of rushed to get patients into the study, so we were oversubscribed, and we ended up with 144 instead of 128 patients. And the dropout rate is much lower than anticipated. I think the physicians like the study, like the drug, and the patients certainly want to stay involved in the study. So this is running now. Because it was 12 months, we built in an interim analysis. So that's coming up soon. The interim analysis occurs when all patients have been treated for 6 months instead of 12. And that time point occurs kind of mid-year. We said publicly that we expect to communicate blinded interim data in early Q3 of this year. And again, it's blinded data. I'll touch on this in the next slide in a minute, but it's a blinded analysis at interim, only looking at safety and biomarkers. And then the final results, all patients are expected to be fully dosed by the end of the year, and we'll communicate top-line results, as we said publicly, early Q1 next year. So it's a very robust study. We did get fast-track designation from the FDA last year. Said fully enrolled. Now it's heads down, execute, and get the data, the interim analysis done, and then the final. So the interim analysis here, it really is a kind of a qualitative look, a blinded analysis, as I mentioned, just to give an indication of safety, which is going to be important, ARIA incidence, side effect profile, and to give an indication of target engagement. You know, we want to show that not only ideally is it safe, but that it's actually it's engaging the target with potential then for beneficial effect or efficacy. So it'll be a qualitative sneak peek, hopefully saying everything's going in the right direction, comes up in early Q3 in a matter of months. There is no clinical analysis at interim. We're not breaking the blind on any of this. So it's important to know that it's a blinded qualitative analysis, as we move forward. The biomarker data, as I said, there's a number of biomarkers that are very well understood in Alzheimer's disease. p-tau217 as a measure of disease progression. GFAP is a measure of neuroinflammation. As I mentioned, the oligomers are highly toxic to synapses. So we've got a presynaptic marker, SNAP25. We have a postsynaptic marker, neurogranin. A-beta 4240 is a true indicator of disease progression as well. So in the blinded analysis, we'll have a few of these data, ideally showing a downward trend in some of these key biomarkers, because if there is a beneficial clinical effect with the drug, we expect to see a downward movement in most of these biomarkers. And importantly for us, kind of over the course of this year, we're certainly not just going to sit on our hands and wait for the outcome. And if the data's there, yippee, now what do we do? So we're plotting and planning really the whole life cycle management for PMN 310. So that not only involves executing on the trial, but what's important is we expect to be more efficacious, so better efficacy, better safety, but we also want to be kind of maximize compliance. So right now, the drug is in the phase 1B trial we're conducting. The drug is administered monthly IV. So our goal is to have a sub-Q formulation to be delivered when we get to commercial launch. So the acceleration, if you will, of the sub-Q formulation will likely kick in later this year. The interim analysis might give us an indication. So to ramp up sub-Q is going to be important to us. The one thing we thought about significantly in the design of the 1B study, that if it's successful, if we can show that it's safe and well tolerated, which we think we can, that enough drug crosses the blood-brain barrier to engage target, that we're actually engaging the target, which will be measured by biomarker movement, and if we have a if we have nailed the dose and we have a clinical signal, that's enough then to step into what we believe is a phase 3 registration study. So in a way, we're kind of bypassing or leapfrogging the phase 2 component and going straight to a phase 3 registration trial on the back of positive data and FDA feedback. But that's kind of the plan as well this year is, and we're in the middle of kind of designing that phase 3 clinical program as well. And then the whole regulatory path, the end of phase 2 meeting, which we'll have early next year once we complete the study. And in addition, the pipeline that I pointed to at the beginning of the study, the next candidates that are kind of coming behind in ALS, targeting misfolded TDP-43, and PMN442, which is looking at misfolded forms of alpha-synuclein for Parkinson's disease and dementia with Lewy bodies. So those pipeline candidates, we want to also march closer to clinic, because ideally what we'd like to say when the Alzheimer's study reads out is that not only have we demonstrated proof of concept in Alzheimer's disease, which will be transformational for the company and ideally for the patients, most importantly, but we'll also show proof of concept of the platform itself. So we want to have those next candidates kind of jockeying for position towards the clinic. So advancing these two candidates, and again, this is pretty cost minimal. We've done a lot of work already on these candidates to move them through preclinical development, but again, marching them closer to the clinic, so then when all this kind of reads out in a matter of months, that people will see that we've got a really robust company focused on neurodegenerative assets with some meaningful, certainly meaningful clinical data. And then last slide then, just as we kind of sum it up, the differentiation, I keep hammering on that. I think it's absolutely important for us. We do believe we have the chance to be best in class for the treatment of Alzheimer's disease. I think there's a lot of focus on Alzheimer's. I think it is going to be a watershed year for Alzheimer's patients and families and caregivers and companies that are successful. And again, if we've got a real chance to lower the side effect issue and come with better efficacy, we'll be off to the races. We do have near-term catalysts, which the investors and the street always likes to see. We've got the interim analysis in a matter of months, and then the final readout kind of 6 months beyond that. And I think the interim analysis will give, as we say here, kind of an early signal on safety and potential in target engagement and efficacy. So that's an important jumping off spot, early Q3. The clinical profile, as I said, the team's done a superb job executing the study and designing it in the first place. We've got our fast track. We're going to get in front of the FDA as quickly as we can to start plotting and planning for that phase 3 registration study. We've been in front of all the strategics. Obviously, there's a lot of interest. There's been a lot of deals done in Alzheimer's disease. I think we've interacted with all of them who are interested to date, so they're all kind of waiting, some at the edge of their seats for the data to come out, so we'll see how that plays out. I'd certainly like to think we'd be in a position ourselves, and again, this speaks to the financial strength. As I said, we just closed on a $175 million pipe with the likes of Wellington and Janus and Ally Bridge, and these guys, deep pockets. And I've gone to them and said, if this trial does read out, I want to be in a position to at least think about funding that phase 3 study ourselves. For me as a CEO, you always want to have options on the table. So coming with a potential pharma deal, which if it goes well, that certainly could be there, but also with the ability to control our own destiny and have a couple of options and weigh them against each other. And these guys that are at the table, have the pockets and have the intelligence to be able to fund that next study. So these are things we're all kind of, preparing and plotting over the course of the next number of months, such that when the data read out towards the end of the year, early next year, that we'll be in a position to kind of react pretty favorably that supports, well, not only the shareholders, obviously, but more importantly, or most importantly, are the patients and the families, because obviously Alzheimer's is a tough one for all of us. So it's if we can do something impactful, that'll be pretty inspirational for us and the team. So that's, as I said, it's been an interesting few months, an interesting year since I stood on this very stage about a year ago. So thanks for your attention. I don't know if you have time for questions. And yeah, thank you.
Brilliant presentation. Congratulations.
Yeah,
Brilliant presentation.
it's okay. You have to speak up a little bit.
Hold it closer to your mouth.
I can, but yeah, it's okay.
You can hear. Brilliant presentation.
Oh, he did say brilliant presentation. I'll repeat the stuff that's important to him.
Congratulations, doing this. One of the questions I have, you did mention in the phase 1A what's the number? How many you're anticipating?
Patient number?
Phase 3 number.
Yeah, so two questions. One around we talked about the drug across the blood-brain barrier. You know, how did we measure that? How do we know how much drug's across the blood-brain barrier? And again, we did lumbar punctures at day 1 and day 29 of that, and these were multiple ascending doses. So lumbar puncture to measure CSF levels of antibody in the CSF, the drug that crossed the blood-brain barrier. And again, it's no different than any other antibody. It was still the 0.1 to 0.3% that naturally traverse the blood-brain barrier. I think the interesting thing in this patient population is that the target itself, the oligomers, are in very low concentration, kind of in picomolar concentration. So it's the amount of drug that we get across, while no different than most antibodies, is more than sufficient to bind the target because the target is so few and far between. And then for the phase 3 design, again, what we're looking at, and there's benchmarks out there, obviously with the Lilly and the ASI trials, and they ran approximately 1,500 or 1,800 patients in their phase 3. So we're fashioning our phase 3 a little bit around those numbers. And we might not have to go as large if our safety profile is much better, which it should be. We can probably ease down on those numbers, but it's going to be 1,000 plus patients. So not a cheap trial, but those are roughly what we've calculated.
Thank you, Neil.
All right, thanks everybody, appreciate it.